Hi everyone, in this video we're going to talk a little bit about antibodies. And antibodies are also called immunoglobulins, and think of these as the proteins that we talked about that could be free-floating in the blood along with other proteins such as hormones and such as albumin and that sort of thing. And they're also, think of them as antigen-binding proteins. So there are 5 types of antibodies, 5 classes of antibodies, and we're just going to talk a little bit about each one just to discuss their major functions. So the first one is IgG, and IgG is the largest class of antibodies. They represent about 80% of all antibodies, and these antibodies can cross the placental barrier. So we mentioned earlier, passive immunity occurs as antibodies cross the placental barrier, and so IgG is definitely the primary example that's going to do this. And then also IgG can provide a defense against viruses, bacteria, and bacterial toxins, and they do so by activating complement, binding to macrophages, and potentiating phagocytosis. IgM is much less prevalent, represents only about 5 to 10% of the circulating antibodies, but they are the first antibody to be produced in an immune response, so they're just sort of the quickest, the most rapidly available. And these are found commonly on B cells, in sort of one form, and then in another form they can be found circulating in like blood and lymph, just unbound, just sort of together. So these guys bind to viruses, and they also bind to foreign red blood cells, and as a result these are the antibodies that are responsible for the cross reactions with transfusion. So if you give a patient B blood who has type A blood and then they have a transfusion reaction, it's generally the IgM antibodies that are responsible for causing the problem. IgA is present in exocrine secretions, so this would be things like tears, mucus, and saliva, and they will attack pathogens before they can even enter body tissues. So they can actually bind to mucosal cells in order to prevent entry of these pathogens. IgE binds to mast cells and basophils, and IgE is primarily responsible for release of histamine. So this is kind of what accelerates inflammation and is one of the major participants in allergic reactions. And then lastly, IgD is probably the least populated of all of them, very, very little of IgD. And these guys actually just bind antigens, and they're generally attached to B cells. So you could think of IgD in the example from the previous video where we talked about how the antigen is engulfed after binding to the antibody on the B cell. That would probably be IgD. So one thing that's interesting to think about in terms of how these antibodies become available, in the case of, we'll just compare quickly the antibody concentration over time based on exposure to particular antigen. So the first exposure to an antigen, so we'll say this is going to be the same antigen either way, is you'll get a peak of IgM, and this would be between like weeks 1 and week 2 of the illness, and then later on you'll get a peak of IgG, so it's a little bit more powerful. You get a little bit more of it, but it takes longer for those antibodies to develop. But if you were to have the same illness again, and I mean this is, I'm not talking about having the cold and then having the cold again, because most likely you're experiencing a different form of the virus, maybe it mutated. But if it was the exact same antigen a second time, we're actually going to see that the response to the exposure is much more rapid and much more powerful, especially in the case of IgG. So we get a faster peak. We get a significantly higher concentration of antibodies. I think that's really nice about this second and any subsequent exposure after the second exposure is that we actually can sort of maintain these memory cells that are developed for years and years and years. So even like 20 years later, if you get the same exact exposure, we've already had one before, you still potentially have the ability to protect against it. All right, and the last thing I want to talk a little bit about in terms of antibodies is what the antibodies can do. So their actions, their functions, their targets, that sort of thing. So we'll just list a couple of them. So number 1, we have neutralization. So an antibody can bind to an antigen on the antigenic determinant site so that that particular antigen can't bind to a body cell. So if we sort of block its its ability to communicate with the other cell, then it can't bind to a body cell and therefore can't cause a problem in that body cell. Second, we have agglutination and precipitation, and these can be classified as 2 or 1, but generally an antibody is like a Y shape, and it can bind to multiple antigens at the same time. So it can actually bind to 2 antigens, and then another antibody could bind to that one and another one. And what we essentially get is this process called agglutination, which is like clumping of the antibodies and the antigens. And the clumps get so big that they actually fall out of solution. And as they fall out of solution, we classify that as precipitation. And so the sort of subsequent response, number 3, is phagocytosis, is that when we have these insoluble clumps, they actually attract phagocytes, and it's easier for the phagocytes to eat them when they're not soluble in solution. So if they're insoluble, then it's easier for them to sort of recognize and consume these clumps that we want to get rid of. And then lastly, we have activation of complement. And we mentioned complement as one of the nonspecific defenses, but we'll just sort of draw it again and just remember that that antibodies are one of the means by which we can activate this system. So the antibodies will bind to, say, like a bacterial cell, and then ultimately the cascade of events that is the complement cascade will cause insertion of the complement proteins into the membrane. And we'll get the membrane attack complexes. We get pores in the membrane, which in the same way as perforins, we can have now free flow of water and stuff in and out of the cell, which ultimately is going to result in cell lysis. So all in all, antibodies are pretty interesting. They actually, think of them as these little tiny things that just have the ability to provide a significant amount of protection for the body. All right, so that is it for antibodies, and that is about it for the lymphatic system. So I hope you enjoyed it. Thanks for watching.